What is SELEX technique and uses of SELEX? Let's get start. SELEX, systematic evolution of ligands by exponential enrichment, is a technique used to identify and isolate specific sequences of DNA or RNA that bind to a particular target molecule. It is a powerful method that can be used to identify and isolate specific nucleic acid sequences that bind to proteins, small molecules, and other biomolecules. The SELEX process begins with a pool of random DNA or RNA sequences. This pool is then exposed to a target molecule, such as a protein or small molecule. The sequences that bind to the target molecule are then isolated and amplified by PCR. This process is repeated multiple times, with each iteration called a round of selection. With each round, the number of sequences that bind to the target molecule increases, while the number of non-binding sequences decreases. This is known as exponential enrichment, and allows for the identification of specific sequences that bind to the target molecule with high specificity and affinity. SELEX can be used for a variety of applications such as identifying AP tamers, which are single-stranded nucleic acid molecules that can bind to a specific target molecule with high specificity and affinity, similar to antibodies. Developing diagnostic assays to detect specific molecules or pathogens in a sample. Developing therapeutics, such as AP tamers that can be used to target specific molecules in the body, such as cancer cells. One example of the use of SELEX is the identification of AP tamers that bind to the protein thrombin, which plays a key role in blood clotting. Researchers used SELEX to identify and isolate specific RNA sequences that bind to thrombin, which were then used to develop AP tamers that could be used as therapeutics to target thrombin in the treatment of blood clots. Overall, SELEX is a powerful technique that allows for the identification and isolation of specific nucleic acid sequences that bind to a particular target molecule with high specificity and affinity. It has many potential applications in areas such as diagnostics, therapeutics, and basic research.